I wanted to start by thanking Susanna for giving me an opportunity to talk about some research that I did during my postdoc in Livermore. And I wanted to start just with this picture of global chlorophyll concentrations. If you heard earlier today, Chris's talk showed some much prettier animated versions of these. I was jealous. But I just wanted to put this up here to show that, primary production, I'm interested in primary producers. A lot of these, when you look at a global chlorophyll map, a lot of this is microbial primary producers. And we were interested in looking at microbial primary producers, and in particular, cyanobacteria. And in general, when you think about these primary producers, you think of them as fixing carbon and putting it in the dissolved organic matter pool, and then it becomes available to microbes. And through this, you sort of get microbial carbon cycling all over the globe. So they contribute to this dissolved organic matter pool. But what we don't often think about is that for cyanobacteria in particular, and other microbial primary producers, the arrow can go the other way. And these cyanobacteria can be consumers of their own dissolved organic matter. And we know that many cyanobacteria can consume glucose, amino acids, all different kinds of organic matter. But what we don't often consider is sort of the ecological context for this and what this means for a complex microbial community when your primary producer is taking up this, changing this dissolved organic matter pool. So we were trying to ask some questions about that and ask some questions about what changes in the dissolved organic matter pool. And to do this, we turned to a sort of model biofilm community that we developed. We pulled out a filamentous marine cyanobacterium from a microbial mat. And with it came some friends, which is a very sort of simplified community. So these associated microbes came along. They end up being about 1 to 5% of the biomass in these communities. The cyanobacteria is by far the majority of the biomass. And what's nice is they form these biofilms that have this complex extracellular organic matter. So this is a picture of it here, stained with an Alcian blue stain that stains the polysaccharides. So you can see this kind of slime gets formed. That's all the blue stuff in there. And then if you look closer, you can see this is just a DAPI stain where you can see the associated microbes. And it's a very low complexity community. There's only about 5 bacterial classes in the associated microbes, along with the cyanobacteria. The first thing we wanted to do was just sort of characterize what was in this extracellular organic matter. And that was some earlier work we did. And we found it's mostly polysaccharides, carbohydrates. And if we look at these closer, mostly glucose-based monomers that are alpha and beta-linked glucose. So you have something that's kind of cellulose-like and something that's sort of starch-like. And there's some other monomers in there, but that's the majority. And then we also looked at the proteins and looked at the extracellular proteins. So that's sort of the next most abundant class of organic matter in this extracellular fraction. And there were a lot of enzymes and transport-binding proteins and proteins of unknown function. And what we found in this sort of first work was that the cyanobacteria were taking up their own extracellular organic matter. And we could see this both in their degradation enzymes that they were secreting and in, through isotope tracing mechanisms. But we wanted to dig into this a little deeper and try and figure out how and why they were assimilating this extracellular organic matter and sort of how that was affecting the rest of the community as well. And to try and answer some of these questions, we used a combination of stable isotope tracing and metaproteomics and enzyme activities to kind of get at the why, the metabolic foundations of why these cyanobacteria were doing this. And for the isotope tracing, at Livermore, we're lucky enough to have an instrument called a nanosims. A nanosims is a high-resolution imaging mass spectrometer that allows you to image to the nanometer scale, so you can image individual cells, and it allows you to get an isotope map of those cells. So you can tell apart, say, carbon-13 from carbon-12, or nitrogen-15 from nitrogen-14. And what this allows you to do is you can make a substrate that you've labeled with either 13C or 15N that you can then trace into individual cells. So I did this with, I labeled just bulk extracellular organic matter that was produced by these cyanobacteria. So they secreted this extracellular organic matter, I collected it, and then I fed it back to a community that hadn't seen 13C or 15N. So this was a tracer. And then you can use nanosims to get an isotope map of the cells. So this is actually an SEM image from this community. These filaments here are the cyanobacteria, and then the little dots here are the bacteria. And this is an image that comes from the nanosims for 13C. And you can see the enrichment, the isotope. This is sort of the EPS stuff that's really enriched, and then you have the bacterial cells and the cyanobacterial filaments. So you can actually quantify how much of this isotope label was incorporated by these individual cells. And so sort of the questions we were trying to answer with this was just to look at something very simple, which is what happens when you turn out the lights. So cyanobacteria are photosynthetic. Obviously, you turn out the lights, you're essentially carbon starving this system. I have not supplied any exogenous organic carbon to this system. So all the carbon is coming through photosynthesis. So when you turn out the lights, you're carbon starving them. And then conversely, when the lights are kept on longer than expected, you're giving a sort of carbon replete system. So we were trying to just compare these very simple scenarios and understand where the carbon went. So I'm starting with daytime. What happens when the lights are turned on for a long period of time? What happens to all this extracellular organic matter? So first of all, the cyanobacteria are assimilating this organic carbon in the light. Let me just orient you with this graph here. This is a graph of all of our nanosims data from individual time points. So we collect individual cells, analyze them with the nanosims, and then sort of aggregate them and get average levels. And what you have here is 13C enrichment and 15N enrichment. And this dotted line is the substrate. So this is what I fed, the extracellular organic matter that I fed the cyanobacteria in the biofilms. And then this point here is what happens in the light. This is the killed control for reference. So they're taking up a lot of this, assimilating a lot of this extracellular organic matter. Because they're above this dotted line, that means they're taking up more carbon than nitrogen from the substrate. They're preferentially assimilating carbon from the substrate. However, this isn't taking into account sort of something very important that I'm showing here in this graph. Cyanobacteria are photosynthetic. In the light, they're also incorporating inorganic carbon. And when you feed this extracellular organic matter substrate that's labeled, you have heterotrophs that can also break down this organic matter and respire that labeled 13C. And then this could also go into the cyanobacteria. So this point here is measuring both this arrow and this arrow. So to try to account for that, we use an inhibitor that inhibits photosynthesis. It's called DCMU. It blocks photosystem 2 electron transport and is blocking DIC assimilation. So with that, what you see is these are all the cells that we analyzed under that treatment. And on average, they go below this dotted line. So what that means is in the daylight, they're absolutely incorporating organic matter, but they're preferentially taking out nitrogen from that organic matter. So it seems like they're taking up organic matter. They're taking up both the DIC. So this is actually also interesting. They're preferentially taking up DIC from this organic matter that we fed them. And then they're also taking up about 25% of their carbon is coming directly from organic matter in the light. And then the other 50% is getting taken up by the heterotrophs. To try and see if we could get some sort of more understanding of why these cyanobacteria are taking up this nitrogen-rich material, we turned to proteome. So this is the total proteome. We also looked at the extracellular proteomes for these organisms under light and dark conditions and compared the treatment. So this is all the red are proteins that went up in the dark and all the light, blue are proteins that went up in the light. And what we see is we see evidence in the light for iron transport, phosphonate transport, amino acid transport. So these are clues to us that in the light, this nitrogen-rich uptake, it might be driven, they might be taking up this organic matter to basically try and scavenge other nutrients that they might need, which makes sense. So how about in the dark? So, oh, I forgot I had one more slide in here. So that's what's going on in the light. This is just sort of a general sort of elemental flux that you can get. You can see that they're actually getting quite a bit of carbon from this extracellular organic matter that's sort of cycling internally in this biofilm. Even though they do have access, this is like grown with plenty of headspace. There's plenty of carbon available from CO2 and bicarbonate in this system. But they're taking about a tenth of their carbon from the DIC and another tenth from organic matter. And they're also competing pretty well, even in the light, with the heterotrophs. And I think this is a really important point. These cyanobacteria can be good competitors for this organic matter. And in the light, at least, it seems to be to acquire micronutrients. In the dark, I don't have time to show sort of all of this data, but in the dark we see a peak of organic matter incorporation after about 6 hours, and then it declines. And to us, this seems to imply that they're quickly taking up this organic matter, metabolizing it, and secreting some metabolic byproducts. And again, in the dark, they're actually even more sort of competitive with the heterotrophs. They take up about 40% of that organic matter. As compared to the heterotrophs taking up 60%. So it seems like they're rapidly utilizing these carbon stores when they need them in a carbon-starved system. And again, we turn to the proteome to see if we can get some more evidence for this. And this is from the extracellular proteomics work we were able to identify some really interesting proteins that were secreted preferentially in the dark. And one in particular that I want to discuss is this carboxymethylene butanolidase that we found. This was a highly abundant exoenzyme that increased in the dark. It's predicted to be involved in other bacterial genomes, to be involved with chloroaromatic degradation, and it has carboxyl esterase activity. And we were able to do some structural modeling and identify active sites, so we were pretty sure we had a carboxyl esterase activity. So then we just turned to enzyme activity assays and looked in the extracellular fraction, and we're able to find that this bulk enzyme activity did increase in the dark. So here we have sort of a paired system where we're able to take evidence we have from the nanosims, go to proteomics and say, look, we have these enzymes, and then go to activity and say, look, we have a way that the cyanobacteria are degrading these extracellular reserves to try and get this carbon. Interestingly, just as a sort of side note, we're also, of course, interested in the heterotroph population and what they're doing. And that wasn't necessarily the focus of these experiments, but one area we're interested in taking this is the heterotroph transporter expression. So transport proteins were some of the most abundant proteins expressed in this system. And we were able to detect all these heterotroph transporters. And interestingly, they did also seem to respond to this sort of light-dark shifts. And there was even a chloroaromatic transporter in one of our representative gamma proteobacteria that was upregulated in the dark. So it seems like they're responding to some of these proteins that the cyanobacteria are secreting and some of these reserves too. And there's definitely some competition going on in here for these extracellular reserves. So finally, just to summarize here, we have a really interesting sort of model system for studying organic matter cycling in sort of a simplified system. And we find, first of all, that the cyanobacteria don't just produce this extracellular organic matter, but they're also consuming it. And they're preferentially consuming it. And it's very dependent on their light-dark status. And so understanding the primary producer's sort of metabolic needs and physiology is really important to understanding the dissolved organic matter composition and pool and its turnover. And there also is some really interesting stories between sort of cyanobacteria heterotroph interactions going on and the heterotroph sort of respiration feeding the cyanobacteria. Okay. With that, I'd like to thank funding from DOE's OBER office and JGI CSP, and also all of my collaborators, everyone at Livermore, NASA Ames, Brad Bebout's lab grows microbial mats and has been sort of necessary for a lot of this project. And all the proteomics work was done as part of PNNL Panomics program with Mary Lipton. And thank you. I'll take questions.
We have a question over here.
Great talk. I was curious about how the heterotroph composition is structured and if that would change with a unicellular population. Kind of curious if there's a selection for specific types of heterotrophs.
Yeah, so yeah, I didn't have time. I put that on a backup slide. I don't think I have it on here. But yeah, so we did look at, we did some ITAG runs in the light and the dark. And so the community is very simple. There's only 5 bacterial classes. There's about 18 OTUs in the whole biofilm system. So it's not a very complex community. So it did change significantly in the light and the dark. We had different communities after 3 days sort of light, dark stress. It doesn't change, it takes about 3 days for the community to change in that respect. And you get 2 populations, the gamma proteobacteria, specifically one OTU, and another alpha proteobacteria population sort of increase in the dark, and then the cyanobacteria OTUs decrease in relative abundance in the dark, which you would sort of expect. Did that answer your question?
And whether, so I was curious about light-dark cycles and whether some of the dramatic changes that you saw in the utilization of extracellular carbon might not be observed in plants that anticipate, or cells that anticipate changes in the availability of light.
Right, so I definitely was thinking about light-dark cycles. So these guys were all acclimated to a 12-12 light-dark cycle. And then, so I think a lot of the data that, a lot of what I'm seeing is so the dark conditions started where they expected the lights to be turned on, and I left the lights off, and then did these isotope tracer experiments. So I think there definitely is some circadian, I was giving them sort of a shock by either leaving the lights on or turning the lights off. And I didn't have time to sort of show but I compared it to a diel where I'd left everything the same, where I had the lights off and on at the, when you would expect, and there's definitely differences, so yes.
All right, one more from Jeff, and then
Oh.
Nice work. So cyanobacteria produce specific intracellular carbon and nitrogen storage molecules.
Right.
So do you think they're producing specific extracellular molecules that they can then more rapidly take up than other bacteria?
That's sort of what we've been trying to get to, just that. So yes, we also do see in this strain, there is this cyanophysin and cyanophycinases that get turned on in the whole proteome. But I think there are some extracellular, what we were trying to find with sort of structures of the EPS when we did sort of that first work was, and what I think we have with the carboxymethylene butanolidase are some specific compounds that are maybe not, the heterotroph population are not able to degrade. So they can leave out these reserves and take them back up. I don't think I have the evidence to say that for sure, but that is the working hypothesis, yeah.